Hello viewers, welcome to my channel. Today we will discuss about the microbiology question paper of GATE XL 2022. To understand the answers, some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. First question, the terminal acceptor of electron during anaerobic respiration in Methanococcus is option 1 nitrate ion, option 2 sulfate ion, option 3 carbon dioxide, and option 4 oxygen. Correct answer is carbon dioxide. The terminal acceptor of electron during aerobic respiration is oxygen, whereas the terminal acceptor of electron during the anaerobic respiration is nitrite, sulfate, carbon dioxide, acetate, etc. Methanococcus is a genus of Methanogenes. The Methanogenes are archaebacteria that produce methane as a metabolic byproduct. Methanococcus is strictly anaerobic and gets its energy by the reduction of carbon dioxide with H2 to generate methane. The Methanogenes use carbon dioxide and acetate as the electron acceptor, whereas they use hydrogen as the electron donor. The next question. Which of the following mutagens convert DNase adenine to hypoxanthine? Option 1 ultraviolet light, option 2 mitomycin C, option 3 methyl methane sulfonate, and option 4 is nitrous acid. The correct answer is nitrous acid. Nitrous acid convert the DNase adenine to hypoxanthine. The pairing behavior of hypoxanthine is like that of guanine. So the AT pairing is replaced with GC pairing. The effect of ultraviolet light, mitomycin C, methyl methane sulfonate is mentioned here. Mitomycin C inhibits DNA synthesis and cross-link DNA at the N6 position of adenine and the O6 and N2 positions of guanine. In addition, single-stranded breakage of DNA is caused by reduced mitomycin C. This can be prevented by free radical scavenger. Whereas the methyl methane sulfonate is an alkylating agent. It acts on DNA by methylating guanine and adenine bases. The UV radiation causes the formation of mutagenic and cytogenic cyclobutane pyrimidine dimers, 6,4 photoproducts, as well as DNA strands breaks. Next question. Which one of the following leukocytes are present in the largest proportion in healthy human blood? This is also MCQ type question. Option 1 neutrophil, option 2 eosinophil, option 3 basophil, and option 4 is monocyte. And the correct answer is neutrophil. That is, among the leukocytes, the neutrophils are present in the largest proportion in healthy human blood. Neutrophil also the first responder to microbial infection. Neutrophils are unable to renew their lysosomes and die after they have phagocytosed a few pathogens. The proportion of leukocytes from high number to low number is mentioned here. This is first is the neutrophil, that is roughly 60% to 70%. That is followed by lymphocytes, that is roughly 20% to 30%. Followed by monocytes, that is approximately 1 to 6%. Followed by eosinophil, that is 1 to 3% approximately. And last is basophils, that is less than 1%. Monocytes are the largest type of leukocytes and share phagocytosis duties with neutrophils. Next question. The site of photosynthesis in cyanobacteria is option 1 chloroplast, option B chromoplast, option C thylakoids, and option D is chlorosome. The correct answer is thylakoid. Cyanobacteria use photosynthetic pigments such as carotenoids, phycobilins, and various forms of chlorophylls, which absorb energy from light. Unlike heterotrophic prokaryotes, cyanobacteria have their internal membrane. These are flattened sacs called thylakoids, where the photosynthesis is performed. The chloroplast is the membrane-bound organelle, also known as plastid, that conduct the photosynthesis mostly in plants and algal cells. But chloroplast is not the site of photosynthesis in case of cyanobacteria. Chlorosome is a photosynthetic antenna complex found in green sulfur bacteria and green filamentous anoxygenic phototrophs, whereas chromoplasts are pigmented plastid, responsible for the pigment synthesis. They provide a variety of color to plant cells and are found in fruits, flower, roots, etc. Next question. The antibacterial microbial activity of vancomycin is due to the option A, inhibition of nucleic acid synthesis, option B, damage to the cytoplasmic membrane, option C, inhibition of cell wall synthesis, and option D is regulation of DNA supercoiling. The correct answer is inhibition of cell wall synthesis. Vancomycin is a unique glycopeptide that is structurally unrelated to any currently available antibiotic. It also has a unique mode of action. It inhibits the second stage of cell wall synthesis of susceptible bacteria. Quinolones and fluoroquinolones helps in the inhibition of nucleic acid synthesis, whereas polymyxins damage the cytoplasmic membrane. Quinolone also regulate the DNA supercoiling by inhibiting the DNA gyrase and DNA topoisomerase. Next question. The phenolic acts as disinfectant by option A, rupturing the plasma membrane followed by leakage of cellular contents. Option B, bond formation between adjacent pyrimidine bases. Option C, forming adduct with amino acid and unsaturated fatty acids. And option D, alkylation of protein. The correct answer is rupturing plasma membrane followed by the leakage of cellular content. The disinfectants are the compounds that kill microorganisms such as fungi, viruses, and all kind of microorganisms, but not the endospore. The phenolic compound have a broad spectrum of activity affecting bacteria, fungi, and some viruses. They work by disrupting cell walls and membranes and reacting with proteins and enzymes. Next question. The nonsense suppressor mutation is found in option A, rRNA, option B, tRNA, option C, start codon of mRNA, and option D is stop codon of mRNA. The correct answer is tRNA. Nonsense mutation is a mutation that alters the codon to a termination codon, such as UAA, UAG, and UGA. Whereas nonsense suppressor is a factor which can inhibit the effect of nonsense mutation. Nonsense suppressor can be generally divided into two classes. A mutated tRNA which can bind with a termination codon on mRNA, or it may be a mutation on ribosome, decreasing the effect of a termination codon. It's believed that the nonsense suppressor keep a low concentration in the cell and do not disrupt normal translation most of the time. In addition, many genes do not have only one termination codon, and the cells commonly use occurred codons as the termination signal, whose nonsense suppressor are usually inefficient. Next question. Choose the correct match for structural component of bacteria for their function. Here in the right side is structural component and in the left side is the function. The structural component flagella, cell wall, metachromatic granules, magnetomes are mentioned here. And in case of function, prevention of cell lysis, chemotaxis, storage for ATP, cell orientation is mentioned. So, flagella helps in chemotaxis, whereas cell wall prevents cell lysis, provides cell shape, and also contain various metabolic enzymes for respiration and another metabolic process. Whereas metachromatic granules are also known as volutins, that are storage reservoir of ATP. That is, for metachromatic granules is option 3, stored of ATP. Whereas magnetosomes are magnetotactic granules that helps in cell orientation or magnetotactic movement. So option S is function is 4. The answer is like P2, that is flagella in helps in chemotaxis and also in movement. Q is 1, that is cell wall prevents cell lysis. Metachromatic granules are 3, that is metachromatic granules helps in storage of ATP, and magnetosomes helps in cell orientation, that is S is 4. Next, match the pathogen with the appropriate disease. In the right side is pathogen, and in the left sides various disease is mentioned. The pathogens are P is Streptococcus pyogenes, Q is Brucella species, R is Mycobacterium tuberculosis, S is Escherichia coli. And that for disease, option 1 is scarlet fever, 2 is Pott's disease, 3 is traveler's diarrhea, 4 is undulent fever. First, you have to choose that which one is correct for the option. As you all, we are well aware about the fact that E. coli is associated with traveler's diarrhea. That is, S is 3. Whereas Streptococcus pyogenes is causes the streptococcal scarlet fever toxin produced that causes the scarlet fever. That is, P is 1. And Brucella species causes undulent fever, Q 4. And R is Mycobacterium tuberculosis that cause the Pott's disease. So the answer is P is equal to 1, Q is 4, R is 2, R Mycobacterium tuberculosis, that is Pott's disease, S Escherichia coli is 3, that is traveler's diarrhea. So Streptococcus pyogenes causes scarlet fever, Brucella species called undulent fever, Mycobacterium tuberculosis causes Pott's disease, and E. coli causes traveler's diarrhea. Next question. Which of the following diseases are caused by the family of DNA viruses? Option A is hepatitis B, option B smallpox, option C influenza, option D is rabies. And the correct answer is hepatitis B or smallpox. Both are consist of double-stranded DNA. Contain double-stranded DNA. That both family are family of DNA viruses. So it is a MSQ type question. Whereas the influenza and rabies are negative sense single-stranded RNA viruses. The next question. Which of the following gram-positive cocci are found in biofilm of dental plaque? Option 1 gonococcus, option B Streptococcus mutans, option C Streptococcus subrenas, option D Fusobacterium species. And the correct answer is Streptococcus mutans or Streptococcus subrenas. Both option is correct. So it is also a MSQ type question. Streptococcus, Staphylococcus, all are gram-positive bacteria. Dental plaque forms sticky film on teeth when bacteria in the mouth mix with sugary or starchy food. Next question. Which of the following statements are true for archaea? Option A is cell wall in archaea contains muric acid and D amino acid. This option is just correct because cell wall in archaea contains pseudomuric acid and L amino acid. Option B is N-formylmethionine is the first amino acid to initiate new polypeptide chain synthesis in archaea. This is also false statement because the first amino acid to initiate new polypeptide chain synthesis in archaea is methionine. Option C is methionine is the first amino acid used during protein synthesis in archaea. This is true statement. And option D is membrane of archaea contains phytanyl rather fatty acids. This statement is also correct. So here two options, option C and option D, both are correct. That is, methionine is the first amino acid used during protein synthesis in archaea, or membrane of archaea contains phytanyl rather than fatty acids. These both options are correct. So it is also a MSQ type question. The cell wall in archaea contains pseudomuric acid and L amino acid. The methionine is the first amino acid used during protein synthesis in archaea. The membrane of archaea contains phytanyl rather than fatty acids. We will further discuss more questions of GATE XL Microbiology 2022 in my upcoming video. Thank you. Please like, share and subscribe to this channel.